Put 2 fingers against the side of your neck, just under the corner of your jaw. Press gently. Feel the pulse. That little knock under your fingertip, about 70 times a minute if you are in reasonable health, is being delivered to roughly 3 pounds of tissue that, by every measure we have, does more computation per gram than any machine ever built. Now, imagine the pulse stops. What I want to tell you for the next half hour or so is what happens inside your skull in the seconds, minutes, and hours after that rhythm goes silent. Because the lights do not go out the way most people think. There is a sequence. There is a wave. There is even, and this is the part nobody told me when I was a young man, a final burst of activity that, by every measurable signature we know how to look for, looks like consciousness intensifying just as the body lets go. I want to be careful, and I want to be honest. So let me start where any honest lecture has to start. With the anatomy. 3 pounds, suspended in a bath of cerebrospinal fluid, behind the bony vault of the cranium, packed like a piece of delicate equipment in shipping foam. 86 billion neurons in there. That count, by the way, is not the old one. You may have learned 100 billion at school. Susanna Herculano Hausel at Vanderbilt University took human brains apart and counted properly for the first time. 86 billion. We were off by 14 billion neurons for decades, and nobody had bothered to check. Each neuron connects, on average, to about 7,000 others. Multiply those out and you get something on the order of 100 trillion connections in your head. 100 trillion. The number of stars in 1,000 galaxies, packed into a vessel about the size of 2 clenched fists held together. Here is what most people do not appreciate about this organ. It is hungry. The brain is roughly 2% of your body mass and consumes 20% of the oxygen you breathe, 20% of the glucose you eat. It is the most metabolically expensive tissue you carry per gram by a very wide margin. Why so hungry? Because every one of those 86 billion cells is doing work, every second of every day. Each neuron maintains an electrical voltage across its outer membrane, about 70 millivolts, with the inside negative relative to the outside. To hold that voltage steady, the cell pumps ions. Sodium out, potassium in, calcium out. And to run those pumps, it burns a small molecule called adenosine triphosphate, ATP. ATP is the currency every living cell uses. Every contraction of every muscle, every memory you can recall, every word you are hearing right now runs on ATP. And here is the engineering choice I want you to notice. The brain holds almost none of it in reserve. The most important organ in your body, the one running the entire show, has a fuel tank that holds only seconds of operation, not minutes. Seconds. Everything I am about to tell you follows from that one fact. One more thing about the anatomy, because the geometry matters. The brain is not one organ. It is several, stacked in evolutionary order. At the base, the brainstem, breathing, heart rate, the autonomic stuff that does not consult you. On top of that, the cerebellum, the motor coordinator. Then the limbic structures, amygdala, hippocampus, thalamus, emotion, memory, the relay station for the senses. And then, draped over the top, the cortex, 3 to 4 millimeters thick, folded so densely that if you flattened it out, it would cover an area roughly 2.5 square feet. The cortex is where you live. The cortex is where the pattern is densest. And the cortex of all of these structures is the most vulnerable to a loss of oxygen. Top to bottom. Newest first. That is the order in which a person leaves. So, the heart stops. Let us say it is sudden. A clot in the left anterior descending artery. A ventricular fibrillation. An electrical fault in the cardiac conduction system. The pump fails. Blood pressure starts to fall. Within 5 to 10 seconds, the brain, perched at the top of the circulatory tree, loses adequate perfusion. Whatever oxygen is dissolved in the blood still sitting in the cerebral vessels gets used up fast. The neurons keep firing. The app keeps getting burned. No new fuel is arriving. Watch what fails first. It is not language. It is not memory. It is not recognition of faces. Those structures take longer. The first thing to go is the integration. The thing that ties all those firing neurons into a single experiencing hold. Around 6 to 12 seconds after cerebral blood flow stops, conscious awareness collapses. Pilots in centrifuge experiments know this exact timing. When sustained G-forces empty the brain of blood, awareness folds in about that window. This is not theory. Aerospace medicine has documented it down to the second. You, the integrated, conscious you, the one listening to me right now, go offline within roughly one breath of cardiac arrest. But the neurons are not dead. The neurons are still firing. The app reserves are still there, just dwindling. What has collapsed is the synchrony. The lights are not off. The orchestra has stopped playing together. Think about what that tells you about the kind of thing consciousness is. If it were stored in some particular cell, in some particular molecule, you would expect a slow fade, a dimming. That is not what happens. What happens is the lights go out in seconds, not because the cells have died, but because the integration has broken. The person over 60 who has stood at the bedside of a loved one having a stroke knows this. Not from the textbook, from the bedside. The eyes are open. The breathing continues. But the look, the recognizing, integrating, this is my husband look, has gone. The hardware is largely intact. The pattern has come apart. And then, for the next several minutes, something happens that almost nobody outside neuroscience knows about. The neurons keep burning their reserve ATP for 30 seconds, maybe 60, maybe 90, depending on cell type and prior conditioning. Some neurons hang on longer than others. The cerebellum, with its tight little granule cells, tolerates oxygen deprivation better than the cortex. The hippocampus is famously vulnerable. The K1 region of the hippocampus, which stores recent memory, begins to die within 4 to 5 minutes. And then the app runs out. When the app runs out, the sodium-potassium pump stops. Remember that pump? The one that uses AP to push sodium out of the cell and potassium in, holding the voltage across the membrane? Without AP, sodium rushes into the cell. Down its concentration gradient. Down its electrical gradient. Both at once. Then chloride follows the sodium, because the inside of the cell is now less negative. Then water follows the chloride, by simple osmosis. The neurons begin to swell, and then, because the voltage across the membrane has collapsed, every voltage-gated channel in every nearby neuron opens. Glutamate, the brain's main excitatory neurotransmitter, floods out. It rushes into other neurons. Those neurons depolarize. They release their own glutamate. The wave spreads. This is called anoxic depolarization. In the older literature, with appropriate drama, it has another name, the wave of death. Anton van Putten in the Netherlands and several other research groups have measured this wave moving across the cortex of dying animals using EEG and direct cortical recording. It travels at a leisurely 2 to 9 millimeters per minute, about the speed of a slow snail across the pages of an open book. You can see it on the recording. It looks like a tsunami of silence, except the silence is not silent. It is the single loudest event a neuron ever produces. A massive terminal depolarization, after which that neuron will never fire again unless rescue arrives within minutes. The wave was first described in a recoverable form called cortical spreading depression by Aristides Leao at Harvard in 1944. The terminal version, the death-specific one, has been confirmed in dying human cortex by Jens Dreyer and colleagues at the Charité Hospital in Berlin, using subdural electrode strips placed during neurosurgery on patients who later died. They published the work in the journal Brain in 2017. We can watch the wave of death cross a human cortex. We have done it. The wave is real. But here is what is strange. The wave of death is not the only signal coming out of the dying brain. There is something else, something the textbooks of my own generation never mentioned, because we did not know to look for it. In 1993, an anesthesiologist running animal experiments noticed something odd on her recordings. When she stopped the oxygen supply to her experimental subjects, the EEG did not simply fade. There was a burst of activity just before the flatline, a burst of fast oscillation. She filed the observation away. Nobody knew what to make of it. 20 years later, Jimo Borjigin at the University of Michigan went back to look. She and her group induced cardiac arrest in laboratory rats and recorded high-density EEG continuously through the dying process. What they found, and published in the Proceedings of the National Academy of Sciences in 2013, was the following. Within 30 seconds of cardiac arrest, in every single rat, there was a transient surge of high-frequency gamma oscillations across the cortex. Gamma waves are the fast rhythms, roughly 25 to 100 hertz, that, in living brains, accompany conscious perception. The moment a thought coheres into something you can hold in your mind, the instant a face you have not seen in 40 years suddenly comes back to you. These gamma waves did not just appear in the dying rats. They became more coherent than they had been in the living animals. The cross-frequency coupling, that is a measure of how slower rhythms in one brain region modulate the timing of faster rhythms in another, increased by a factor of 5 during the dying period. Phase locking between distant cortical regions increased by a factor of 8. These are integration measures. In a living brain, those numbers go up when you are paying close attention to something, when you are having a vivid memory, when the world feels crisp and present. In the dying rats, those numbers went up. They did not go down. They went up. Then the activity collapsed into silence. In 2023, Borjigin and her colleague Gang Shu published a second paper in the same journal. This time the subjects were not rats. They were 4 human patients in the intensive care unit, in coma, with life support being withdrawn at family request. Continuous EEG was already running. 2 of the 4 patients showed the same pattern: a surge of gamma, a surge of long-range cortical coupling, activity rising in the temporoparieto-occipital junction, a cortical region that in living people is implicated in conscious experience, and specifically in the felt sense of being inside one's own body. All of it happening after the heart had stopped. All of it happening as the brain was, by every conventional metric, dying. Borjigin and her group were careful in the discussion of that paper. They did not claim their patients were having a near-death experience. We cannot know that. The patients did not survive to report. But they did say, and this is the line that stayed with me when I read it, that the data are consistent with a hyper-aroused conscious state arising in the dying brain. I want to repeat that, because I think it deserves repeating. The data are consistent with consciousness intensifying in measurable signatures of integration in the seconds after the heart stops. Now, before I tell you what I think this might mean, I have to do the other half of the job. Because the gamma surge is activity. It is not yet evidence of experience. And to think clearly about that gap, you have to think clearly about what consciousness actually is. Consciousness, as far as the best models can tell us, is not a thing. It is not a substance, not an organ, not a glow stored in some particular cell. It is a pattern, a specific kind of integration of information across a single physical system at once. Giulio Tononi at the University of Wisconsin has formalized this in what he calls integrated information theory. Christoph Koch at the Allen Institute has carried the work forward. The theory is mathematical. It is contested in its specifics, but the central idea is one most of its critics accept: that what makes a brain conscious is not the number of neurons, but the way information is integrated across them. The richness of the causal connections, the pattern. You are not 86 billion neurons. You are the pattern those neurons make when they synchronize. I learned this lesson when I was young, though I did not have the words for it then. My first wife, Arline, died of tuberculosis. She was 25 years old, and the thing I remember standing in the room after was, that body lying there is not Arline. That is the carbon, and the calcium, and the hydrogen. The pattern is what is gone. The pattern was everything. So when we ask, what dies? The answer is, the pattern. The atoms remain. Every carbon atom in your brain right now will continue existing for billions of years. Most of them will pass through other creatures, other plants, other oceans, other stars. Carl Sagan said this beautifully. We are made of star stuff, and after we go back to being star stuff. Take a single carbon atom. Right now it is part of a neurotransmitter in a synapse in your hippocampus. It is participating, this very second, in the encoding of whatever it is you are paying attention to. In 6 months it might be in a wheat plant in Kansas. In a decade in a fish in the Pacific. In 1,000,000 years, locked into limestone at the bottom of an ocean basin. In 1,000,000,000 years, given the right tectonic motion, ejected back into space. The atom does not change. The atom does not remember. The atom does not care. What changes is what the atom is participating in. What is lost, then, is the connectome. 100 trillion synapses, each one tuned by decades of your specific experience. The strength of every synaptic connection, the precise wiring of every dendritic branch. That is the recording of every face you have ever loved, every song you have known, every late afternoon when you were 6 years old and the light came through the kitchen window at a particular angle. The synaptic weights are physical structures, but they only have meaning when the electrical activity flows through them. The score requires an orchestra. Without ATP, without the relentless second-by-second pumping of ions against gradients, the music stops. The atoms persist. The pattern dissolves. And the pattern was never made of anything other than ongoing process. There was no separate soul substance underwriting it. There was just this, the continuous, energy-hungry, ATP-burning, ion-pumping dance of 86 billion cells, generating in their synchronized whole the thing you have been calling I. Now I promise to tell you what we know firmly and what we are still guessing about. Let me do that, because the honesty is the whole point of the exercise. What is solid? The metabolic cliff after cardiac arrest, the 6 to 12 second window of consciousness loss, the apt depletion sequence, the anoxic depolarization wave. These have been measured in countless animal preparations and confirmed in human cortex during neurosurgical monitoring. You can put numbers on every step. You can replicate every observation across labs. This is textbook physiology now. Bet on it like you bet on the second law of thermodynamics. What is well supported, but young? Borjigin's gamma surge. The rat data is robust, replicated, large sample, controlled. The human data is, and I want to be honest about this, 4 patients, 2 of whom showed the pattern. That is not nothing. The signature is striking and consistent with what was seen in rats. But it has not been replicated by other labs in large human samples because the experimental conditions are extraordinarily difficult to capture. Patients dying with continuous high-density EEG monitoring already in place are rare. Expect the number to grow. Right now, treat the gamma surge in humans as a real signal worth taking seriously, not yet as a settled fact. What is suggestive but contested? The connection between the gamma surge and reported near-death experiences. Sam Parnia at New Langone runs the AWARE studies, large prospective trials of cardiac arrest survivors. He has documented cases where survivors report awareness of events that took place during their resuscitation, events they could not have seen with their physical eyes. Pim van Lommel, a Dutch cardiologist, published a prospective study in The Lancet in 2001. 344 cardiac arrest survivors. 18% reported some kind of recalled experience. The phenomenology is strikingly consistent across cultures. Whether it represents real perception during a period when the brain should have been incapable of perceiving, or memory reconstruction during the recovery phase, that remains genuinely open. Bet small. What is speculation? Anything I or anyone else might say about what the gamma surge feels like from the inside. We do not know. We cannot know directly. We can map the physics. The phenomenology, the what it is like, is for now beyond instrumentation. If anyone tells you otherwise, count your wallet. There is one more thing I want to point at, because anesthesiologists have known it quietly for 50 years, and medical schools sometimes mention it and sometimes do not. Every general anesthesia you have ever received, every appendectomy, every wisdom tooth, every hip replacement, is a partial, reversible, carefully titrated rehearsal of the molecular cascade I have just described. Propofol, the milky white drug that puts a surgical patient under in 15 seconds, works by enhancing the activity of GABA-A receptors. GABA is the brain's main inhibitory neurotransmitter system. Boosted GABA activity hyperpolarizes neurons. Hyperpolarized neurons are harder to fire. The long-range synchronization between cortical regions degrades. The pattern we call consciousness fragments. Sound familiar? It is the same cascade as anoxic depolarization, just routed through a different intermediate step, and, this is the critical word, reversible. The anesthesiologist holds you at a particular depth. The drug is metabolized. The synchronization returns. You wake up. When we do anesthesia, we are quite literally taking the brain to the edge of the cliff and walking it back. And ketamine. Ketamine is now widely used for treatment-resistant depression, and at higher doses it produces the dissociative experiences sometimes called K-holes. Experiences that phenomenologically resemble the reports from near-death survivors. The British psychiatrist Carl Jansen wrote a careful book about this overlap in 1997. Ketamine blocks NMDA receptors. NMDA receptors are densely involved in the glutamate flood that follows ATP depletion at death. The pharmacology is not identical to dying, but it is, and I want to use this word carefully, adjacent. Here is the dose calibration point I have to make, because the comparison only lands if the numbers are right. A bolus of propofol sufficient to induce general anesthesia in an adult costs about $2 to $5 at hospital wholesale. A single anesthesia event in an American hospital, counting drug, equipment, monitoring, the anesthesiologist's salary, the residency training behind that salary, runs anywhere from $800 to $4,000 depending on procedure complexity. The global anesthesia drug market is roughly $20 billion a year. The body's own version of the cascade, the one that runs when the heart stops, has no provider. It runs on its own pharmacology with its own neurochemicals, and it runs all the way to completion in roughly 6 to 10 minutes if no one intervenes. You should not, under any circumstances, take what I have just told you as a suggestion that there is some pleasant or revealing experience to be sought at the edge of death. The anesthesiologist holds you at the edge with milligrams of drug and tens of thousands of dollars of monitoring equipment and decades of training. The cliff itself is one way. What I am pointing at is something subtler. The mechanisms by which consciousness dissolves at death are not different in kind from the mechanisms by which consciousness is suppressed in surgery every day. The same molecular pathways, the same hyperpolarization, the same loss of integration. Just one is held reversibly, and one is not. We have, in a quiet way, mapped the pharmacology of dying so precisely that we routinely walk to its edge and come back. The people I have loved and lost did not have the luxury of that round trip, but the pharmacology is the same one running in their cells and ours. So, take your fingers off your pulse. Let me close where I should close. The atoms in your body have been around for billions of years. Almost every heavy element, the carbon in your DNA, the iron in your blood, the calcium in your bones, was forged in the core of a star that exploded before the sun was born. You are a temporary arrangement of very old material. When the arrangement ends, the material returns to circulation. Some of it will be in a tree in 100 years. Some of it in another person in 1,000. Some of it, given long enough, will be in the next star. That is conservation of matter. That is physics. What is not conserved is the pattern. The pattern that is you, the 86 billion cell synchronized dance of ions and glutamate and gamma waves and reconstructed sensory experience. That is a process. Processes are not stored. Processes only exist while they are running. My mother used to say of someone who had died that they lived on in the people who loved them. I used to think that was a soft kind of consolation. A polite thing, one says. But I want to tell you what that sentence actually means in the physics. The pattern that was her was not stored anywhere only inside her. It was distributed. Every interaction she had with another person, Every conversation, every gesture, every story she repeated, every habit she taught without meaning to, left a small change in the synaptic weights of the person across from her. When she died, her own copy of the pattern dissolved. But the partial copies, the distributed echoes, the shape she had pressed into the people who loved her, those were still running. On other hardware. That is not a metaphor. That is the actual physics of social memory. The pattern is preserved, partially, in distributed form. She lives on, in the only way a pattern can. So, the thing my mother said, and the thing the physics says, are the same thing in two languages. The heart stops. The wave crosses the cortex. The gamma rises. The synchrony collapses. The atoms go back into circulation. And the pattern, the specific arrangement that someone has spent a lifetime pressing into the people around them, continues to run, in pieces, for as long as those people continue to run. That is what we have when someone is gone. We have the copies they left in us. We have the gestures we make without thinking that we got from them. We have the way we hold a cup, the phrases we use without remembering where we picked them up, the silences we have learned to keep. The pattern is not nothing. The pattern is what survives. Tell me one specific thing you still do without thinking, that you learned from someone now gone.